Alright. Siapa yang I'm here tu? Betul Haris. Layanan tak ada eh? Tell him I miss him, okay? Okay, in the name of Allah, the most gracious and merciful. Okay, actually renal physiology, as we teach the undergraduates, this is the system that has most lecture slots. It comprises of 12 lecture slots, so contributed to around 12 hours of lecture. So bear with me. I think I've done the renal countercurrent, the urine concentration, renal handling of water. So part of it is done in the previous session. So I really hope that what I prepared today will really help you to be a good physician, surgeon or physician later. All right. So, all right. This is the learning outcome after studying renal physiology. A student is expected to understand and able to discuss renal clearance, glomerular filtration, and renal blood flow. Okay, this three sounds very I'm not sure scary or simple. Some of you, because this is subjective, yeah. But if you take it this way, these three, renal clearance, glomerular filtration, and renal blood flow, is actually, why do you need to know this? Because this is how you assess the renal function of the patient. That's why you've got to know the renal clearance, because depending on the substance that has been cleared by the kidney, it will tells you the GFR. You can estimate the plasma blood flow and also renal blood flow. That's it. Okay, not just merely a theory that you memorize para amount of uric acid and then inulin and things like that. Think of its application clinically. We are using creatinine, but you have to know the basic. Why you need to know this is basically to test a function of the kidney of your patient. Okay. So next is renal tubular reabsorption and secretion. Okay, this is the most important processes that is occurring in the kidney plus the filtration kan. Filtration, reabsorption and secretion. Just the three processes, right? And then the renin-angiotensin aldosterone system. So this is, I think, bread and butter of renal physiology as well as cardiovascular physiology, because this is very important to maintain the homeostasis of your blood pressure, blood volume and water level in the body. As you all know, we are 70% water. So imagine how important this system is. Yeah. So any student who cannot answer renin-angiotensin aldosterone system, it's not that you cannot be a doctor, but maybe not yet. Okay, you've got to learn, more. This system should be at the tip of your finger. And then as we go on with the function of the renal, okay, it is very important to regulate the level of electrolytes in the body. The most important one that you should know is sodium, because there are so many other solutes that depends on sodium level in the body. For example, water really depends on the sodium concentration inside and outside of the cell. Therefore, there are some electrolyte regulation that you've got to know in detail. For example, sodium and potassium. Yeah, calcium, of course. Because this is an important ions for what do you call that, action potential, right? And also muscle contraction. Okay. And then renal sodium and water regulation. This one has been covered in the previous session, yeah, osmoregulation. How do you maintain sodium homeostasis in the body? The contribution of aldosterone on maintaining the homeostasis of renal sodium and water, and the kidney countercurrent multiplication. And then you've got to be able to explain to us the renal endocrine function, because renal are able to secrete erythropoietin, as well as the helps in the formation of vitamin D, and then acid-base physiology. I think this has been covered by Fatin, right? Specifically focusing on acid-base physiology because kidney is one of the major important organ in regulating acid-base balance, all right? And then its compensatory mechanism and the micturition or urination process. These are all the learning outcomes that we expect the students to know when they learn renal physiology. So far, so good? A lot, right? It's quite a lot, okay? Okay, now I make it simple for you. So looking at the kidney, so Alhamdulillah, everyone is blessed with two kidney. It's a twin organ, bean shape. Ni takkan lupa ni, surgeon mesti ingat kan. So the main function is to filter blood. The other function is to regulate. Okay, this one is 2 marks, 1, 1, 1, 1. So it becomes 10. So if you elaborate, it can becomes 25 lah for your essay. So it helps to regulate the blood pH, blood volume, blood pressure, osmolality, and able to produce hormones. So basically, these are the main functions of the kidney. Okay. Okay, this is just anatomy. If you still remember, you have right and left kidney. Kidney is actually protected by the what they call that, ribs, yeah. And then there's a special structure here called hilum. This is where the entrance and exit for everything, not everything lah, all this, ureter, renal artery, vein, lymphatics, and also nerve. And also renal is a retroperitoneal organ. Okay, it is there behind the peritoneal membrane. So it is covered by first the renal capsule, followed by adipose tissue and the fascia. Okay, so this is the functional structure a little bit. You've got to know that there are cortex and medulla. So cortex are outer cortical and inner juxtamedullary zone here, and there are renal columns in between the pyramids. All right. So these are the pyramids. Okay, are located in the medulla. Okay. The renal medulla comprises of about 10 to 15 renal pyramids with the tip here, which is called as papilla. Okay, and then this one will form the minor calyces, major calyces, renal pelvis, and towards the ureter. All right. Okay, day by day for over 20 years that you have lived in this world, all right, the kidney filter of around 150 liters of blood per day. If let's say the average of us have 5 liters blood in the body, that means the entire blood volume is filtered for 30 times a day. All right, 30 times a day. So the whole blood is being filtered in less than an hour per day, right? So therefore, the kidney receive a lot of percentage from the cardiac output, about 25% of cardiac output from the aorta, it goes to the right and left renal arteries. Right, now you've got to memorize this structure, the functional unit of the kidney, which is this, the nephron. Okay, the nephron, it is comprises of the vascular part, the vessel, and then the tubular part, the tubules, and then the mixed vascular tubular part, which is the juxtaglomerular apparatus. I will show you later. Okay, so for the renal blood flow, if the question is asking for renal blood flow, you've got to explain lah. It comes from the aorta and then to the renal artery, segmental artery, interlobar, arcuate, and then to cortical radiate artery. So from this cortical radiate artery, it goes to the afferent arteriole first. Then this will form a new capillary loops known as glomerulus. So this is capillaries again. And then this one will continue forming arteriole, not venules. Yeah? It becomes arterioles, and these arterioles will form another set of capillary, which is known as peritubular capillary. Peri sebab tepi, tubule, tepi tubule, peritubular capillaries here. Then only these peritubular capillaries will form veins, right? So this is the nephron, the functional structure of the kidney. All right, now we zoom into the renal corpuscle. This one, renal corpuscle comprises of the glomerulus and the Bowman's capsule here. So you see, Bowman's capsule, if you are asked to draw the structure, I expect you to be able to draw the structure. You should have the afferent arterioles, the glomerular about 6 to 8 loops, and then the efferent arterioles here. Okay, and then what is so special about this glomerulus is that it is what do you call that? This is where filtration occurs. Okay, and then if we zoom into the capillary of the glomerulus, you can see that the important structures are, okay, first is the endothelium here. Okay, and then followed by the basement membrane. Okay, the outer part of the capillary, we have the epithelium, which is like interdigitating among each other, yeah. So it is like macam octopus, macam tu lah. Some books explain it like octopus, the what they call interdigitating like this. So this cell is known as podocyte, yeah. So it forms the filtration slits here. So it allows passage of water, glucose, ionic salts, but not allowing large proteins and red blood cell to pass through. So this is the one that contributes to the filtration process in the Bowman's capsule. Okay. Okay, this is again the tubular part. You start with, okay, tubular part, you start with the Bowman capsule here. Lepas tu yang convoluted ni sebab dia convoluted lah. Convoluted, this is proximal. Lepas tu ada lagi convoluted, this is distal. So in between these two, there are loop of Henle, ada descending and ascending. So descending you have thick and thin, ascending you have thin and thick. Why do you need to know this? Because the properties are different. They have different pumps, different protein channels that will alter that will help us to create the concentrated urine. So this is DCT and this is the collecting duct. And this one, urine will be formed through here and then towards the minor calyces. Okay, so the filtrate that we filter will be fine-tuned based on the needs of the body. Alright, so water and solutes will be filtered. So if the body needs it, then we will reabsorb to the peritubular capillary. But if we want to excrete it, so it will be in the filtrate in the renal tubules and will be excreted in the urine. Okay, so now to the second topic. So far so good? Am I too fast? Am I too slow? Ke demam dah? Dah vaksin dah ke semua Are you all vaccinated?
Yes.
Yes. Okay, Alhamdulillah. Okay tak lepas vaksin? Semua kuat-kuat? Okay, ni lagi famous tajuk glomerular filtration rate. You jangan takut. Glomerular, apa yang filtration, apa yang filter dekat glomerular, rate ni mesti over time. So apa yang boleh filter? Of course the plasma, right? The fluid being filtered at glomerulus, rate ni per unit time. So kita guna minute lah. So remember the definition. Nanti masa Viva jangan takut. Kalau orang tanya, just think of the words. You will get the answer. Okay, in textbook, it is 125 ml per liter per minute for average 1.73 meter squared. Normal range is 90 to 120, but I think very rarely for us not a nephrologist to do this, right? Okay. So what are the factors? The factors affecting the glomerular filtration rate. What makes it increase? What makes it decrease? This is a favorite question in physiology. Okay, so glomerular filtration rate actually it depends on the net filtration pressure. If you remember net filtration pressure, of course, it's a hydrostatic pressure and oncotic pressure, kan? Through the Bowman capsule and capillary. Okay, the different between the oncotic and the hydrostatic pressure, both inside the capillary and the Bowman's capsule, will contribute to the net filtration pressure. So this net filtration pressure will depend on your blood pressure and the proteins that are available in the blood. So that contributes to net filtration pressure. So nanti jangan takut lah, you cakap je lah oncotic pressure in the capillary, hydrostatic pressure in the capillary, and the oncotic and hydrostatic pressure in the Bowman's capsule. But of course, in the Bowman's capsule, you do not have the oncotic pressure, right? Because that by right, the proteins is negligible. So you have a positive net filtration pressure favoring filtration from capillary to the mana? Filter from mana? Bowman capsule to capillary ke capillary to Bowman's capsule? Yes, and then forming filtrate. Okay. So other factors is the what do you call that? The constant. Constant. This one is constant is not change in normal individual, but in a disease patient, this one can change. Property of the glomerular membrane denoted by glomerular surface area. Like say for a patient who has diseases affecting the what do you call that? The glomerulus or the meningeal cells, so it will affect the surface area as well as the permeability of the glomerular membrane. Permeability ni sometimes loss of charge and things like that. So by right in a normal person, this one is not changed. Tapi in disease, it can be changed, which is the glomerular surface area and also the permeability of the glomerular membrane. So these are the factors affecting glomerular filtration rate. So whenever the glomerular surface area decrease, of course GFR will be decreased and vice versa. Okay, so far so good. GFR, you need to know this because you just because you want to know whether the patient's kidney is functioning well or not. Yeah. Alright, how do we regulate GFR? Okay, regulation ni GFR more or less the same lah as the renal blood flow. And also it depends on the blood pressure regulation. Intrinsic is the renal auto-regulation. That means the local regulation of blood flow inside the kidney. But extrinsic is the systemic blood flow regulation, which will also give impact to the GFR. Alright, so intrinsic, okay, we call it as renal auto-regulation and local regulation. We have the myogenic theory and the tubulo-glomerular feedback theory, whereby myogenic theory, it is operated by stretch receptor. The more you, if there's too much stretch, so it will constrict. That's how it controls the blood flow. Locally, let's say that the blood flow is reduced, so therefore it is not stretched, then it will dilate, so to allow more blood flow, right? Tubular glomerular feedback theory, I will explain to you later. Okay, extrinsic and normal blood regulation, we have the autonomic nervous system from the neural control, and we have the hormonal control, which is the renin angiotensin aldosterone system. Which will also affecting the ADH, aldosterone, and the other one is the adrenal natriuretic peptide, which is which has the receptor inside the heart. Okay, tubular glomerular feedback mechanism. Okay, this is tubular glomerular feedback mechanism that's actually an intrinsic control of regulation in the blood flow, whereby it is detecting the flow through the renal tubule, the flow through the renal tubule, and it will communicate the JJ cell, the granular cell here in the afferent tubules. So what happens if, let's say, the GFR is reduced, therefore there will be reduced flow through the medullary cells. So this will initiate the paracrine from medullary densa to afferent arterial, causing dilation in the afferent arterioles. Therefore, will increase the hydrostatic pressure in the glomerulus to increase GFR back to normal. Other than that, this medullary densa will also stimulate the JJ cell to release renin, causing activation of the renin angiotensin aldosterone system. But the glomerular feedback is the what you call that local effect here. Yeah? Renin, angiotensin 2. Therefore, this angiotensin 2 can will also cause constriction in the efferent arterioles, leading to increased hydrostatic pressure in the glomerulus, thus increase in the GFR. So there are two ways. Yeah? So what does it mean by paracrine mechanism here? So for example, this one is reduced GFR. Let's say there are increased renal blood flow, there will be increased delivery of sodium and chloride to the macula densa cell here, right? So this sodium and chloride will enter via sodium potassium chloride co-transporter at the DCT here. So there will be increased sodium potassium ATPase activity and ATP hydrolysis that release increase adenosine release. Therefore, there will be increase of calcium ion, leads to vascular smooth muscle contraction in the afferent arterioles here. Therefore, it will help to decrease GFR. Tapi if the renal blood flow is low, so this process is vice versa. Therefore, vascular smooth muscle will vasodilate and GFR will increase. You do not need to remember this. This is not at your level. This is your level. This is for physiologists, yeah? The basic mechanism of it. But if some of you would like to know how this paracrine, so this is the explanation. But you do not need to remember this. Okay? Okay, now we move to clearance. So far so good, yes. Now I have finished with renal blood flow and GFR. I'm moving to renal clearance. So far so good?
Yes.
Okay, good. All right. So, okay, you just need to know some of these substance lah, which is discussed in the textbook. Albumin, not filtered langsung. Glucose memang filtered tapi completely reabsorbed. So this is glucose. You just imagine the diagram tu kan. If it is not filtered at all, for example, it is albumin. Kalau it is filtered but completely reabsorbed, example is the glucose. Kalau freely filtered across the membrane, neither reabsorbed nor secreted. So these are inulin. So this will helps you. This will tells you the GFR. But we are using creatinine kan, because this inulin is not readily available in the body. So when we use creatinine, dia tak panggil GFR lah, kita panggil estimate GFR. So if you note, there will be small e over here, eGFR. That means to say that one is using the creatinine lah, estimates GFR. Okay. If let's say para-aminohippuric acid, so this is renal plasma flow marker. So both filtered and secreted, not reabsorbed. So these are para-aminohippuric acid because we are doing this study because we want to know the renal plasma flow. So if you want to know the renal blood flow, you have to consider the hemoglobin. There are special calculation, right? So you can check back on that. So these are the substance, at least 4 substance that you've got to know. Okay, that's all for renal clearance. Renal handling of sodium, I'm not sure whether you need to know the details of this. Let me skip this video first. I think I can give you this video later for you to see at home. Okay, talking about renal handling, it's actually how the nephron, what is the other word for handles? What is the fate of sodium when it is being filtered from the glomerulus? So if you're asked about renal handling, that means you're going to say whether it is filtered or not. What happens in the glomerulus? And then you tell us what happens in the proximal convoluted tubule. Is it reabsorbed? Is it secreted? Is it what do you call that, reabsorbed and secreted lah, either reabsorbed or secreted or none. Okay, and then what happened at the loop of Henle, descending, ascending, and then the DCT and CD. So what's happening? So for sodium, it is important because mainly the pump is the sodium potassium ATPase pump, right? So this one is actually explained as well in the renal countercurrent mechanism. Yeah, renal handling of sodium. You can watch a video lah of this. If I have time, I will show you. Okay. Okay, now juxtaglomerular apparatus. Okay, what is juxtaglomerular apparatus? Juxta means close to, if I'm not mistaken, close to or beside, next to, something like that, glomerular apparatus. So juxtaglomerular means next to glomerulus. So this is the glomerulus, this is the juxtaglomerular apparatus. So just next to the glomerulus, okay. So this is such a busy slide. Apa ni? Okay, the main component of this juxtaglomerular complex or juxtaglomerular apparatus is the first one is the JG cell, juxtaglomerular cell here. This is actually a modified smooth muscle cell from the afferent arteriole. Okay, can you appreciate this? Are you with me?
Yes, Doktor.
Yes, okay. So if you are asked to draw about JJ apparatus, you should draw the glomerulus. Kena ada afferent arterioles ni, afferent arterioles. Kena ada distal convoluted tubule ni. Right? So this one you draw the afferent arterioles and then you draw the granular cell ni, color hijau. Okay, this is the juxtaglomerular cell. These are the cells that secrete renin. Not glomerulus, not medullary cell, not messenger cell, not macular densa cell. So the granular cell or the JG cell, this one is the one who secrete renin. So all my students have to know this. Okay, and then you have the macular densa cell, blue color ni. So this is a modified chemoreceptor cells from distal convoluted tubule cell. Right. The next important one is the extraglomerular mesangial cell here. Alright, extraglomerular mesangial cell. So these three is the important component making up the juxtaglomerular complex. Okay. Okay, the juxtaglomerular cell receive signal. It will receive multiple signal, alright, in order to secrete renin. It sends low blood pressure. So what stimulates renin release is actually renal artery hypotension or low blood pressure. Okay, this will stimulate the renin, the release of renin by the juxtaglomerular cell. Okay, the macular densa cell, it is a chemoreceptor kan. This one is a mechanoreceptor. This one is a chemoreceptor sensing sodium and chloride level. So it will be activated when it sends low sodium and chloride delivery to the distal convoluted tubule here. So there are two guards here lah, at the entrance and at the exit. We are still monitoring in the kidney to ensure our blood volume and blood pressure is sufficient to sustain life. Right? And then these extraglomerular messenger cells helps in the communication between macular densa and the juxtaglomerular cell. It helps in the signal communication between macular densa and the juxtaglomerular cells. Alright, so this JJ apparatus actually helps to regulate blood pressure, glomerular filtration rate, and controlling by means of blood going through the glomeruli each minute. Okay, now towards the end of my lecture, renin-angiotensin-aldosterone system. Okay, as you all know, the cardiovascular system ni sebenarnya is a pump je kan, dia macam pump lah, heart ni memang pump, so it keeps on pumping the blood. Tapi who maintains the amount of water, amounts of red blood cell, amount of electrolytes is not the heart, it's the kidney. Kidney is the organ that will what do you call that? That will that perform the action whereby if you need, yes, batuk ke? Tak. Are you okay? You all gelak-gelak, you all sama-sama satu komputer ke lain-lain tempat ni?
Lain-lain.
You all dekat SOPD? Seminar room. Seronok lah. You all kan boleh kerja. You all essential kan? I kan tak. Okay. So the kidney is the one who is doing the job to remove water if needed, to maintain water if needed. The cardiovascular system, dia pump je. Tapi the heart, as you know, is the, this one is what Prof. Azarisman told me lah. The heart is the most, jujur. Apa jujur dia panggil? Trustworthy, is it? Paling jujur. It pumps blood but it never steals any oxygen from the blood that it pumps. It waits for the, it only receives oxygen from the coronary artery, and this is the very first organ that functions in our life. It starts on functioning right from the 6th week when we are inside our mom's womb, right? I'm not saying that it is not important in this matter lah. I'm saying this is a pump, but the renal who is actually controlling the amount of the water, electrolytes, and also erythrocytes. Alright. Okay, so the blood pressure, satu pressure tau, satu lagi volume. When you explain in the Viva or when you answer your question, you be very careful whether you are focusing on the pressure or volume. Okay, so this is tightly controlled under the system called as renin, angiotensin, aldosterone system. Right. So this is just repetition lah when you okay, the blood flow from renal artery to smaller arteries, afferent arterioles, and then to efferent arterioles, glomerulus, efferent arterioles, peritubular capillaries, then to the veins. What is this here? Oh, this is to the veins. All right. Okay. Okay, what is the stimulus to renal release? I told you earlier, right? Okay, the first signal is the mechanoreceptor, which is the granular cell here, the juxtaglomerular cell, which is located next to the glomerulus in the afferent arterioles. Yeah? So it signals that the blood volume needs to be increased. That means to say it responds to renal artery hypotension. Okay, this is known as also baroreceptor lah, because it is a mechanoreceptor. It mechanically feel low blood pressure. So whenever the blood pressure is high, so this cell will inhibit renin release. Alright, so whenever the blood pressure is low, so the blood vessel collapse, the arterioles collapse. So this will stimulate this Jg cell to secrete renin. So that's how it functions, because it's a mechanoreceptor. So that is the renin release by signal number 1. Okay, signal number 2, as I told you, the stimulus is also from the autonomic nervous system, which is the sympathetic nervous system. Yeah. So this actually the receptor is also a mechanoreceptor. Okay, baroreceptor located in the carotid sinus and also aortic arch. Yeah, the baroreceptor, it measures the heart, it measures the blood pressure from inside the heart. So this actually has a connection to the granular cell in the afferent arterioles. Alright. So whenever this vessel is stretched, alright, so this sympathetic nerve fiber will be down regulated. Therefore, renin will not be released. Whenever this vessel is collapsed, so the mechanoreceptor will sense this and sympathetic nerve fiber will be upregulated and this will stimulate the granular cell. So how does it stimulate the granular cell? Is by the presence of beta-1 adrenergic receptor at the JG cell. So this beta-1 adrenergic receptor will be stimulated as in response to low blood pressure. So renin will be secreted by the JG cell whenever the sympathetic nervous system is activated. Okay, this is number 2. How do you promote renin release? Number 3, by the chemoreceptor from the macula densa cell here, the distal convoluted tubule. Okay, sense when glomerular filtration increase or decrease, it detects the quantity of sodium and chloride ions. Okay, if let's say there are reduction in the chloride and sodium ion, therefore renal artery hypotension, this will send signal through the mesangial cell to the juxtaglomerular cell to release renin. So these are the 3 ways of how the granular cell is stimulated to secrete renin. So far so good. Only 3 stimulus. Okay? Yes, so far so good. Okay.
Yes.
Okay. What will renin do? So renin ni dia bukan duduk dekat kidney lah. Dia akan masuk dalam capillary. Okay. And then it will cleave. Ini macam sword tau. Macam pedang ke pisau, whatever you call it lah. So this renin in the capillary, it will cleave angiotensinogen. Angiotensinogen ni is a large protein. Dia about 400 kita panggil apa 400 amino acid. Ni 8 amino acid kan. This is about 400 amino acid. So renin dia akan cleave angiotensinogen. Jadilah angiotensin 1. Angiotensin 1 ni I think is around 10 amino acid. So of a smaller size. Okay, so angiotensin 1 being cleaved from angiotensinogen which is produced by the liver, right? So this angiotensin 1 will circulate in the capillaries and then there will be converted to angiotensin 2 by angiotensin converting enzyme that is readily available in many types of capillaries but most abundant in the pulmonary capillary. Okay, so this angiotensin converting enzyme will cleave angiotensin 1 to the cleave 2 molecules becomes angiotensin 2, a smaller protein which contains only 8 amino acids, lagi kecil lah angiotensin 2. So there is angiotensin 2. So what will this angiotensin 2 do? Okay, so as you all know, angiotensin 2 is a very potent vasoconstrictor. Yeah, it affects almost all smooth muscle, almost all peripheral smooth muscle in the arterial. Okay, but in the kidney, it is predominantly in efferent arterial. You are the afferent, you are the efferent kan. Because if you, let's say you constrict your afferent arterial, you will have reduced GFR. Agree? Constrict afferent. Agree? Okay. If let's say afferent is normal, tapi efferent ni you constrict, GFR increase ke decrease? Increase because dekat sini hydrostatic pressure dia increase lah bila you constrict at this part, kan? So that's why as a postgraduate level you should know that the effect of angiotensin 2 is more predominant in the efferent arterioles. Okay, tapi when the angiotensin 2 level is so high, it's very high, it will constrict both efferent and afferent arterioles. Therefore, it will reduce GFR lah because you want to increase the blood pressure, right? So if let's say the level is so high, then both arterioles will be constricted. Therefore, GFR will be reduced. Therefore, will increase your blood pressure. but in normal circumstances, it is the efferent arterial which are more responsive rather than afferent arterioles, yeah, towards the action of angiotensin 2. So not only affecting the total peripheral resistance in the kidney itself, it helps to preserve water, yeah, angiotensin 2. That's the first two effects that you should know. Okay, this is what I explained before. Okay. Now, what happened at the proximal tubule? Tadi dekat afferent and efferent arterioles, right? So at proximal convoluted tubule, PCT, right after the glomerulus here, angiotensin 2, it will increase the reabsorption of sodium. Reabsorption of sodium into the cell, so water will follow. So this part at the PCT, actually angiotensin 2 will increase the reabsorption of water and sodium, therefore it will help to increase your BP. So that is the third action of angiotensin 2. Right? And then next, angiotensin 2 will also affect hypothalamus. Alright? And this will, what do you call that? It will stimulate the thirst center to increase the your fluid intake, right? And then it will stimulate the release of antidiuretic hormone. So when this antidiuretic hormone is released, so what does it do is that it will affect the what do you call that? This part, collecting duct. There will be a lot of insertion of aquaporin water channel at the luminal side at the collecting duct here. Therefore, this aquaporin will enable water reabsorption from the lumen, for the tubular lumen back into the cell, then to the peritubular capillary. So more water will be reabsorbed because of release of antidiuretic hormone from the pituitary. All right. Okay, as you all know, the blood pressure depends on the total circulating blood volume, the cardiac output times total peripheral resistance. So therefore, when there are increased thirst, there will be increased oral intake, increased water reabsorption, increased blood volume. So this will increase the total circulating blood volume. And then because of the systemic peripheral vasoconstriction, so there will be increase in total peripheral resistance or increased peripheral arterial resistance leads to increased total body resistance. So therefore, this will increase the blood pressure. So this is how the renin-angiotensin aldosterone system mechanism helps to increase the blood pressure. Okay, the other effect of angiotensin 2 is that it will affect the apa nama ni? Gland apa ni? What gland is this?
Adrenal.
Yes, adrenal gland. Apa-apa?
Adrenal gland.
Pandai. Apa Hafizuddin yang jawab? Adrenal gland. So which part of adrenal gland? There are many layers like ada zona glomerulosa, fasciculata, reticularis. So dia akan stimulate the zona glomerulosa to secrete aldosterone. So aldosterone is actually a mineralocorticoid that will regulate sodium balance. Tapi sodium ni of course it will affect water because water will always follow movement of sodium. So another effect of angiotensin 2 is that it will stimulate the adrenal gland to secrete aldosterone. Okay, what will aldosterone do? Aldosterone ni actually is I can't remember. Dia bukan directly to the protein channel. It's actually stimulating, bind to aldosterone receptor complex. This will control gene expression. So there will be gene transcription, transcription of a new protein. So this new protein, this process will take hours to days. Yeah. So this is a late response of renin-angiotensin aldosterone system, whereby gene expression will cause the insertion of sodium potassium ATPase pump at the basolateral membrane in the late DCT and also CD over here. Okay, so as a result, what do you call that? There will be increased reabsorption of sodium in exchange with potassium. Okay, as a what do you call that? As a response to increased aldosterone release. So sodium will be reabsorbed to the blood, potassium will be exchanged over here. Therefore, there will be sodium reabsorption from the tubular lumen and potassium will be excreted out in the urine. So, intense activation of renin-angiotensin-aldosterone system may result in hypokalemia. Okay. So water, as usual, will follow sodium. Sodium gets reabsorbed here. So actually, it is active process here by sodium potassium ATPase pump. Then when the sodium concentration is low, sodium from the tubular lumen will enter here via the sodium apa channel sini? ENAC kan, ENAC channel over here, and then it will be reabsorbed back to the circulation. So water will follow. So this will boost the blood pressure up. Okay, this is how the effect of angiotensin 2 and aldosterone causing increase of the blood pressure. Okay, in summary, the renin-angiotensin aldosterone system. Apa ni ya? Okay, ni dia punya stimulus is detected by the baroreceptor both in the granular cell as well as the carotid sinus and aortic arch. Okay, the baroreceptor, and then it is also detected by stimulating and signaling through the activation of sympathetic nervous system and also macular densa cell, which is a chemoreceptor detecting the low sodium and chloride ion in the tubules, causing the activation of renin, causing the JG cell to secrete renin, which will cleave angiotensin 1 from angiotensinogen, and then angiotensin converting enzyme will cleave angiotensin 2 from angiotensin 1, and this angiotensin 2 will exert its effects on the efferent and afferent arterioles to actually, when you want to increase the blood pressure, you have to reduce the glomerular filtration rate. Tapi kalau minimum, not very high level of angiotensin 2, it can increase the glomerular filtration rate. This is the local response of the kidney. And then it will affect the proximal tubule to reabsorb more sodium. It will cause increased thirst, to increase fluid volume intake, and also increase the blood pressure to the insertion of aquaporin water channel for reabsorption of water, and also aldosterone that will enhance the reabsorption and retention of sodium and water, thus promoting the increment of blood pressure. Okay. Okay, I think, ni ada micturition. Micturition ni I think senang je kan. Okay, these are the structure of the kidney. Then you've got to just to tell you that, okay lah, from the kidney, the urine is formed and then will traverse to ureter towards the bladder. If you cross section the bladder, you will see the uretrovesical junction here. Okay, kalau the bladder is full, this uretrovesical junction will help to will compress the opening of ureter, so kita prevent blood flow of urine. So urinary bladder ada special rugae. This can stand the pressure that is increasing in the bladder without causing us the urge of urine. Beside that, we have this detrusor muscle that helps with the contraction. And then we have this special trigon over here, the sensitive area to stretch. Okay, I think you all surgeon tahu kan benda ni. And then, okay, the what do you call that? This is so crowded in female, but quite free in male. Therefore, this will cause the ability to hold urine, which is lesser in female, especially during pregnancy, is even worse because of the crowded structure here in the in relation to the bladder. So by right, we can hold around 750 mils, but of course lesser in a woman. Okay, trigon sensitive to expansion. So this area is actually signals us when it's time to pee. So yang ni tak apalah, urethra I think you know kan the different parts of urethra. Okay, now the maturation reflex. Maturation reflex ni dia ada dua center tau. Satu maturation center dekat S2, S3 level kat sini. Satu lagi dekat pontine level. Dia ada maturation center dekat sacral here and also pontine. Kalau pontine ada dua, pontine storage center that stops maturation reflex and pontine maturation center that allows the reflex for us to pee. So this maturation reflex, it depends on the contraction of the bladder. So usually if the what do you call that? If the urine or the bladder is half full, about 300 to 400 ml, we will have the feeling to pee. So there will be a reflex sent to the maturation center. This center will cause this signal will cause a reflex whereby this maturation reflex causing the detrusor muscle to contract. Bila detrusor muscle ni contract, it will cause the mechanical opening of the internal sphincter. Sphincter over here. This is the skeletal muscle. This skeletal muscle is actually contributed by the pelvic muscle. So this is under voluntary control from the higher center, from the cortex. So this is where that we can hold the urine. But this area, it is operated by the maturation reflex at the sacral level. So this is just a recap lah. Stretching dia ada spinal cord, and then from pontine, how do you, for example, kalau pontine storage, this center actually allow or inhibit maturation. Kalau pontine storage center is activated, it will inhibit maturation by acting on all three spinal efferent. It will inhibit parasympathetic activity, increase sympathetic activity, and increase somatic motor nerve activity. So this will inhibit urination. I think you can go with the diagram lah. This is the sacral. This is the pontine maturation reflex. Okay, that's all. All right, are you good? Okay, any questions? Let me stop sharing.